All right, it looks like it's 12:30. I'm sure we have people that are going to continue logging on for the next few minutes, but I'll get started with introductions. So this is our Creighton School of Medicine virtual research brainstorming session. Thank you all for being here. And this is really meant to pull our community together across the Omaha and the Phoenix campuses, and basic science research and clinicians and learners in a group and start a conversation. So I'll just a little bit of an update on our schedule. Today we have Dr. Jason Bartz, a professor in the Department of Medical Microbiology and Immunology and chair of that department, and also Associate Dean for Faculty and Academic Affairs here in the school. And I'll introduce him in just a minute, but before we get to our speaker for today, the calendar is up. Our next session will be September 22nd, focused on autism, with a group of both basic scientists and clinicians talking about a couple different projects related to that area. And then October 27th, infectious disease and antimicrobial research, and Dr. Nancy Hansen's presenting. And it looks like also somebody from ID here in Omaha will present as well from the clinical side. So I'm delighted to introduce Dr. Bartz as our speaker. His research has won awards and is very well supported by the NIH, groundbreaking research into prion diseases. So I think this is a great opportunity for us to hear about ongoing and developing project and start a conversation. So I think that the presentation itself is not that long, and we only have one speaker today, so we have plenty of time for questions. I would imagine, I don't know, Dr. Bartz, do you want people to hold their questions to the end, put them in the chat? Nope, they just
have questions. The less I talk, the better.
All right, then the floor is yours. Thank you so much for being here and doing this today.
No, thank you. So I think what I'd like to do is talk about some of our work and kind of as an overview. I'm just going to give a very brief background on prions, just because they're kind of this novel infectious agent and they're really weird. And then talk about some of the work in my group, and we do a lot of interdisciplinary work. So I work with environmental engineers and wildlife biologists and neuroscientists. So I'm kind of excited to present in front of this group today. And kind of the overall arching kind of goal of our work and what I'm most interested in is prion evolution. So as I'll talk about in a few slides, prions are these protein-only infectious agents, and they can evolve. And I'm really interested in just understanding, how does a protein-only agent respond to new replication environments, be that a new host, in response to anti-prion drugs, or the environment. And that's kind of what I'm going to talk about here. And kind of these three main areas of work in my group, studying the fate of prions in the environment, modes of infection, and kind of emergence of prions, all really kind of are interact with each other. And from my point of view, I'm always thinking about this through the lens of prion strains and prion evolution. So prion diseases, so in the classic prion diseases, these are diseases of humans and in animals. And humans are Kuru of the 40 people of Papua New Guinea, Gerstmann-Strasser syndrome, fatal familial insomnia, and CJD. Prion diseases in humans are unique in biology in that they can have three etiologies. They can be a sporadic disease, they can be transmitted by an infectious agent, and they also can have a familial etiology. Prion diseases of animals include scrapie of sheep and goats, transmissible mink encephalopathy of ranch-raised mink, bovine spongiform encephalopathy or mad cow disease, The most recently identified prion disease is camel prion disease. And then the one that's gaining the most attention right now and is chronic wasting disease. And so CWD occurs in both captive and free-range cervids, so white-tailed deer, mule deer, elk, moose. It's been identified in Nordic countries and reindeer and red deer. Has an expanding geographical range. This is a map. This is the most recent map of the CWD distribution from July of this year. And you can see it is fairly widespread throughout North America, including Nebraska. It has an increasing incidence. So in areas, there's some areas in Canada and in the United States where over 60% of the deer tested test positive for CWD, which has a negative impact on herd health. And as I'll talk about in just a little bit, just again, due to the protein-only nature of the agent, is that the prions are pretty remarkable, is that they can persist in the environment for decades. And it's thought that they can bind to soil and they can be taken up by plants. And this persistence in the environment is thought to help spread CWD in natural settings. The host range is poorly defined. So again, one of my interests is interspecies transmission. But there's experimental evidence suggesting that CWD can transmit to sympatric species such as swine and cattle. And it's estimated in the US about 15,000 people a year consume, either knowingly or not, CWD infected deer. So we have kind of a giant interspecies transmission experiment going on in the US right now. So the prion agent, again, as I mentioned, this is unique in biology. It's this protein-only infectious agent. And so historically, these amyloid deposits that occur in the CNS of infected people and animals, if you purify the deposits from the brains, excuse me, these deposits are comprised of these long linear non-branching amyloid fibrils, and then biochemical analysis of the fibrils indicates that they're comprised of this single protein called the prion protein, which is encoded by the host. And this data led Stan Prusner to propose that this protein itself was the infectious agent. He happened to be right and received the Nobel Prize in 1997 for this work. So the prion agent is again, it's really a disease of protein misfolding. So the normal form of the protein, or PRPC, and the C stands for cellular. This is expressed in all of us. Pictured here is an NMR structure on the left, high alpha helical content, sensitive to proteases, detergent soluble, really don't know what it does, but through a post-translational conformational change, it undergoes a kind of wide-scaled conformational change from this alpha helical content until about 2 years ago, I could just say beta sheet, but recently the chromogram structure of PRPSC has been solved and has a parallel and register intermolecular beta sheet structure or PIR structure. It's highly resistant to proteolytic degradation, insoluble detergent, and it's the infectious agent. It doesn't need anything else but protein. So if you're a protein-only infectious agent, how do you make more of yourself? So in the literature, and I kind of mistakenly use replication because I think it's a poor way to think about this. So prion conversion is probably, I think, a better way, in that you have the prion form, or PRPSC, interacts with the normal form, and through a process that we just don't understand, somehow it redirects the conformation of PRPC into SC. You get these amyloid fibrils that grow in the cell, and these fibrils then, again, through a process we don't understand, are sheared, creating new friends that can feed into the system, and you get this exponential increase in prion formation. It's also an interesting way of explaining the 3 different etiologies of human prion diseases. Again, you have this infectious etiology where you're putting the preformed fibril into the host, for example, in Kuru via cannibalism, or in the spontaneous and familial, there's lots of experimental evidence that the familial forms, these are all mutations in the prion protein, and the spontaneous is that you get the spontaneous conversion of the normal form into the infectious self-templating conformation. And it's the one example that I'm aware of that Pasteur was wrong, is here you get the de novo creation of infectivity. So one of the things I'm really interested in, and kind of actually it was one of the best arguments against the prion hypothesis back in the day when they're arguing, oh, was it a virus or a prion, is just there's prion strains. So just like every other infectious agent, you can have different strains of prions, and these are operationally defined as a phenotype of disease under a fixed set of agent and host parameters. And the phenotype can be differences in clinical signs, in incubation period, neuropathology, and then the distribution of the agent and the host. And then also something that I'm really interested in is that the zoonotic potential of different strains can also differ. And this is work from my thesis advisor, just showing that in rodent systems of prions, they can have very different incubation periods, very different clinical signs of disease. And so,
sorry,
yeah.
can I ask you, but the same sequence?
But the same sequence, yeah.
Wow.
And that great question, because it segues into the next
Crazy.
slide, is that so it's like, well, how is this? And so it was hypothesized that you had the same amino acid sequence, but different conformations. And the conformation was what encoded the strain properties. And again, up until last year, I would just say it's all indirect evidence. So things like differential migration on Western blots, or relative sensitivity to proteolytic degradation, or if you do this assay called conformational stability, where you basically melt the protein and ask how much denaturant does it take to unfold it. Those were strain-specific properties. But now with the cryo-EM, is that a group in the UK has solved the cryo-EM structures. And these are two mouse-adapted prion strains. They have different incubation periods, different neuropathologies. They have the same amino acid sequence, but they have different structures. They're both pure structures, but they do differ. So this is the most direct evidence that, again, there's different conformations, same amino acid sequence, different conformations. Now kind of, I think the next step is a structure function analysis. Is we know the structure, but how do these structures encode the phenotype of disease? And I think that's one of the things that my group is really interested in, is trying to understand these structure function relationships.
May I chime in?
Yeah.
So then when you say conformation, I'd rather say the different morphology of the aggregates, because that within the aggregate, the individual chain seems to be different conformation, but really the whole aggregates, I think, is the infectious agent.
No, indeed. So there's work suggesting you need between 15 and 17 individual monomers to make an infectious unit. So clearly the structure that's shown in the cryo-EM contributes to kind of that different in the aggregate overall morphology. So yes, it is clear it's more than just the tertiary structure. There's something with the quaternary structure. There's probably something to do with fragmentation. I mean, all of these things where we contribute to what we see.
And then may I ask a question correlated to this one? In the Alzheimer's disease, in amyloid beta, long time it was held that the aggregates are the toxic agent, but later it turned out that the soluble oligomers of amyloid beta are the toxic agents. Is there any similarity here?
There is, and so I think there's a common theme, and I'll get that at the end, is that all of these protein misfolding diseases, which are referred to now as like prion-like diseases, so Alzheimer's, Parkinson's, tauopathies, they share a lot of similarities, and one of them is what you're mentioning, is that the toxic species doesn't seem to be these large aggregates. They actually might be protective, but it's the more soluble oligomers that may be the neurotoxic species. So for PRP prions, I think that's becoming increasingly clear as well.
Okay. Thank you.
No, of course. So that's kind of the very short introduction into prions, and then just kind of, again, a very high level of what my group is working on. And again, I can really kind of break it down into three components, is our work with the environmental engineers and civil engineers on the fate of prions in the environment, work with kind of neuroscientists and microbiologists on modes of infection, and then a lot of work on prion strains and kind of this idea of prion evolution. And so for the environment and work, this started with a collaboration with Shannon Bartell-Hunt. Shannon is the chair of civil engineering at the University of Nebraska-Lincoln. And we have worked together for about 15 years on this, and it's really been productive and super interesting. I got to learn a whole, well, I know very little about engineering, so this is why you collaborate with engineers. But what we found is we just started asking basic questions that it's known that prions can persist in the environment for decades, and just kind of how is that? And we started asking very basic questions about, we know that prions are shed into the environment, and so can they bind soil? And kind of what we found is through a series of papers is that yes, prions can bind soil, and it was way more complicated than I could ever imagine. So it's dependent upon the soil type. So things like clay bind prions much more avidly compared to things like sand. It is dependent on prion strain. So if you change the strains can bind to soils with different affinities. The matrix that contains the prions also effects of prions are shed in urine versus blood versus a carcass. That affects exactly how prions interact with the soil. And then once prions are bound to soil, again, all of these kind of parameters determine how infectious it is. And so we have a whole series of paper examining how prions, once they bind to particles, how do they retain infectivity? And then kind of in the next series of experiments is how do they respond to weathering? So again, we know prions can survive in the environment for a long time. And so this is work from Chian, who was a graduate student at the time. And what she found is that prions were really extraordinarily resistant to natural weathering processes. So here we did a series of experiments of repeated cycles of drying and wetting, simulating what would happen in the environment, and found that, again, it was strain dependent, and what type of soil it was would affect how this mitigation process would work. Did another series of experiments with freezing and thawing, again, taking advantage of, at the University of Nebraska, is that they have the equipment in the civil engineering to test concrete for repeated freezing and thawing cycles. So we kind of hijack that equipment and ask, well, what happens with prions? And the short answer is you can freeze thaw prions 500 plus times, and they really don't care. And interestingly is that we found that when they're in a dehydrated state, they became, extraordinarily resistant. And we're starting to identify the mechanisms of how they can persist in the environment for such a long time. One of the overarching findings of these studies was that it was also strain dependent. There were certain strains that were more or less resistant to weathering. And so Sarah Holek, who was a graduate student in my lab at the time, who's now a postdoc at UMass Amherst, she found is that if you mix different prion strains together and then simulated these environmental weathering processes, is that you could alter the ratio of the strains and actually drive kind of evolution just by environmental factors. So this is kind of the first evidence in the field that interaction of the prions with the environment could select for certain prion strains. And this was based on kind of the soil type that it bound to, and also the weathering conditions. Someone can imagine in the field with chronic wasting disease, depending on where you are in North America, what your weather is like, what the geology of the area is, that is going to have an influence on what strains may survive the environment and be transmitted to the next host species. So one of the things that we're working on for technology development is how do you detect prions in the environment? This has been historically very difficult to do. And so this is work from Chi again. And what she did is she developed a method where you can swab a surface and extract the material from the swab, concentrate it, and then put it into this technique called RT-Quick. And all I'm going to explain about RT-Quick is that it's analogous to PCR, but for misfolded proteins. So you can take very small levels of PRPSC or the prion and amplify it to levels where you can detect it by conventional methodologies. And what she found is that you can swab surfaces that basically contain a single infectious unit, and we're able to detect prions from there. Now, Sarah Simmons, my group, is carrying this work forward and asking questions about if you disinfect surfaces, can you validate the disinfection processes using this procedure? And what we're trying to do right now is develop like a swipe test for laboratories that work on prion diseases. So when we do our decontamination protocols, that that we can validate that the decontamination is successful. And something I'll talk at the end is that we want to move this into a clinical setting, again, validating that surgical instruments and surgical settings, et cetera, are free of prion or prion-like proteins. So kind of quickly moving into the next step or next part of the work in my group is we're really interested in pathogenesis kind of questions. And so this is work that we've done here with Tony Kincaid. And Tony is a neuroscientist and made the observation is that a lot of the animals that develop prion diseases are macrosomatic, meaning that the nose is kind of how they explore the world. And so we did actually kind of a simple series of experiments showing that following inhalation is compared to ingestion, and ingestion is kind of the way that we think that most prion diseases are transmitted in the wild. But what Tony found and what we found is that inhalation not only caused disease, but it was 10 to 100-fold more efficient compared to ingestion. And so this was described, we described this in hamsters, but then our colleagues at the USDA down the road in Ames replicated this in sheep and held true in sheep. Our colleagues in Zurich did this in mice, worked in Zeig and worked in mice. And then our colleagues at Colorado State did these inhalation experiments in deer, and in fact found that not only did it cause disease, but was more efficient than ingestion. And so a lot of the work that Tony and I have been involved with since this observation is just asking, well, why? Why is this more efficient? And it turns out that things were happening much more quickly than we thought. In the sense of prion diseases, again, slow virus diseases, they'd have very long incubation periods. And it took us a long time to realize that the important events that were happening following inhalation happened within minutes to hours versus what we thought might have been days to weeks. And we found that PRPs seek across the epithelium, get into draining lymphoid tissue or lymphatics within minutes post-infection, and then able to seed lymphoid tissue and cause infection. And then these series of experiments also, again, if we use different strains, and this is work done by Ron Chiki in my group, is that we found that different strains have different efficiencies for being infected via inhalation or ingestion. And again, it's another mechanism of strain selection. And then we kind of dissected the mechanism out. And what we found is that strains like hyper, which can cause disease following inhalation, and strains like this drowsy strain, which don't cause disease, is they both cross epithelia, they both get into draining lymph, they both get into blood, they both can make it to spleen, whereas hyper can establish infection in spleen and then gain access to the peripheral nervous system, but drowsy is unable to establish infection in spleen. And then we had another series of papers kind of exploring the mechanism of why that is. So then we've been focusing on just this part here of what's happening in the nervous system for prions and prion transports. And so Jacob Ayers, who is a graduate student at the time of my group, who's now faculty at UCSF, what he found is that when you inoculate a peripheral nerve, in this case, the sciatic nerve, with different prion strains, which target different areas of the CNS, have different incubation periods, is that they actually all use the same four descending motor tracts. So the transport of prions didn't seem to differ between strains, but what he found is that the rate of spread was strain specific. Unfortunately, the techniques that we had at the time, we couldn't differentiate between the actual inoculum we put in and newly replicated. So that rate of spread was the combination of both replication and the original inoculum. And so Sam Koshi, who's an MD PhD student in my lab right now, has gone through and developed a really powerful technology where we're able to purify prions, label them with a fluorescent tracer, and now do live animal imaging in the sciatic nerve. And what Sam is finding is that these different prion strains actually have the same rates of transport. And the rates of transport that Jacob was describing were consistent with slow axonal transport. And what Sam is finding when he's actually measuring the actual PRPSC molecule transported along the nerve is that they're actually fast axonal transport. So we're kind of, I think Sam's work is kind of changing the paradigm of how we think prions are transported in the nervous system. And then lastly, our work is working on strains. And as I mentioned before, I'm really interested in this idea of how do prions evolve? How do they react to a new replication environment? And this started with interspecies transmission and adaptation. And so it's long been known that when a prion goes from species A to species B, there's a long incubation period, and then subsequent passages, it adapts and becomes more pathogenic for that new host species. And this has, again, long been observed. And it's thought to be a strain selection mechanism. And what we found is that it was a little bit more complicated than that, in the sense that when you would artificially mix different strains together and inoculate them into animals, it wasn't simply a race to the end, but the strains actually interfered with each other. So like a lot of infectious agents, is that the infectious agent is not only kind of competing with the host, trying to evade the immune system, et cetera, but the infectious agent is trying to compete with all the other infectious agents that are also trying to infect that host. And prions are no different that way. So prion strains, they could interfere with each other. And we've spent a long time investigating this mechanism. It seems to be a major driving force in how prions adapt to new host species. And through a very long series of papers, we basically determined that strain interference is governed by the relative onset of prion replication between strains in a common population of cells. The prion strains compete for the normal form of the protein. So if this is in excess, they don't compete. And that the blocking strain can suppress replication, but does not completely eliminate the super infecting strain. I'll say one more thing about this. So the work that we're going forward with this is that we're actually identifying what we're calling substrains. That strains tend to be a very dynamic mixture of kind of a dominant strain and substrains. And we're actually developing techniques now to identify these substrains. And the substrains have different host ranges. And again, we think this is an important mechanism for, again, how strains can quickly adapt to a new replication environment. We've also kind of delved into synthetic biology. So again, prions being this protein-only infectious agent, you can synthesize synthetic prions where you take recombinant PRP, the normal form of it, and you can get it to misfold in the infectious conformation. And Alyssa Block, who recently graduated from my labs, now the postdoc at Colorado State, what she found is that you could make these synthetic prions and recapitulate known species barrier events that have been well described in the field. So we're kind of excited about this in the sense now we have this very trackable system that we can start to modify to maybe get a handle on some of these structure function relationships between the structure of PRP-SC and how it can adapt to a new host species. And then the last and kind of the newest area for our group is again the prion-like diseases, and so this was kind of first suggested again to Stan Prusner about a decade ago. This is from a review in Science where he suggested that other protein misfolding diseases, Parkinson's, Alzheimer's, etc., are all prions in the sense of that they're a disease of protein misfolding, and that the prion form of the protein can bind to the normal form, convert it into the prion form, and that's how it propagates more of itself. And then you get resulting kind of deposition. You have the A beta plaques, tau tangles, Lewy bodies, in the different diseases. And since this was published in 2012, there has been a huge amount of literature now consistent with this hypothesis. This is from a review from a few years ago. Again, what I've talked about is the prion diseases or classical prion diseases, so GSS, FFI, CJD. It's all the same protein, but different conformations give you these different diseases. APP, Alzheimer's disease, ataxin is in these different diseases, Huntington with Huntington disease. And what I want to focus on here is with alpha-synuclein, with Parkinson's disease, multiple systems atrophy, and dementia with Lewy bodies. Again, like what Laura's question, it's the same amino acid sequence, but there's different conformations and it gives different phenotypes of disease. And so this is, from a prion biology, this is strains. And this is how we're kind of thinking about these diseases now. And so this has resulted in a collaboration and an R01 with Amanda Warman, who is works on Parkinson's and multiple systems atrophy, and kind of combining her expertise in Parkinson's and these diseases, and kind of my expertise in prion strain biology. And we're asking questions about tropism and this whole idea that if you have different conformations of alpha-synuclein, how does it affect kind of the spread and targeting within the central nervous system? So it's kind of this combination of, again, Parkinson's disease and prion biology. And I'm kind of fired up about this. I just got back from my first Parkinson's meeting, and I think we have a lot to contribute to the field in the sense of a lot of the paradigms they're talking about Parkinson's disease. I'm thinking about them more as a microbiologist and a prion biologist. They're very similar. It's pretty remarkable. So lastly, it's like, well, how do we kind of expand our collaborations and kind of expand these studies? So things I'm very interested in is, again, applying kind of this prion strain paradigm or the prion paradigm in general to these other prion-like diseases. Kind of a big argument right now in the field is over nomenclature, just because, I don't know, scientists like to argue over nomenclature. And there's a lot of people are really resistant to calling, like Parkinson's, for example, a prion, which I get. But I think from a scientific point of view is that these things like strains, et cetera, I think are very applicable. And I think it's a good way to make testable hypotheses that one can work on. So this is something that I'm starting to engage neurologists here at Creighton. So Melinda Burnett, she's been joining some of our journal clubs. And then I guess the second area is this novel technology we've developed for detecting prions in environments. And this is, again, based on some of the work that Amanda did when she was a postdoc in Prusner's lab, is that she and now others have followed this is multiple system atrophy. Again, this is alpha-synuclein, is that they exhibit stability and resistance to inactivation. So basically applying what we know in the prion field, things like formalin fixation, heat inactivation, is that the activity of those MSA aggregates to cause disease in transgenic mice is not inactivated by these common disinfection procedures. And there was just a recent paper that came out showing that dementia with Lewy bodies will survive autoclaving, et cetera. Again, very reminiscent of what we've known in the prion field for 30 years. And so I'm interested in, again, expanding our detection technology that we can do with PRP prions, and then using this for alpha-synuclein and A-beta for making sure that surgical instruments, et cetera, medical devices are actually decontaminated. All right, so that's all I have. Just thank all the people that do all the work. I just talk, and they're the ones that actually do the stuff. And so, yeah, any questions or?
Really nice. That was really interesting. I'm sure deafening applause behind all those mute signs. I don't know how we applaud on a Zoom, but there you go, Anne. Thanks, Jason. Okay, so open up. I have my own questions, but I'll wait. If you want to unmute and jump in, anybody?
I saw someone wave their hand. If
Oh.
not, I can go.
Go
Yeah,
ahead, Anne.
this is Anne O'Keefe. I'm just thinking about this with an epidemiology hat, and wondering, how much information do we have about cases of these, prion diseases and prion-like diseases where we could look at different risk factors, and wondering, if maybe some of these quote unquote spontaneous diseases are actually acquired in the environment, the strain that might have evolved in the environment or something like that.
Yeah, no, that's a really good question. And I was at a meeting this spring and some individuals from the CDC were presenting data from areas that have very high incidence of CWD, asking if there's a corresponding increased incidence of human prion disease. And as of right now, there is not. But the CDC admitted, part of the difficulty is that, as you know, there's so few autopsies that a lot of these diseases get misdiagnosed. So it's very difficult to, apply some of these epidemiological tools just because, the data set that you have is not totally reliable.
Yeah, and they may not even be reportable diseases.
Correct.
So, yeah,
Correct.
very interesting.
I love the way that it's so clearly bridging over into translational and clinical applications. I mean, it's really beautiful work. So I guess I'm wondering about the treatment side specifically. So things like the half-life of the normal PRPC or other prion normal versions, and thinking about, I mean, it's hard in the nervous system, but thinking about, it seems like, if there's no known function for the normal protein, then a knockout animal should be completely resistant, right?
Yep.
So, and then thinking about CRISPR-Cas9 editing, is there a way that we can, I mean, I would imagine for like Huntington's families, being able to do editing in that population with, their high risk, that could you edit away the disease susceptibility, or if protein turnover is fast enough of the normal protein, could you edit enough of the protein that, of the gene, so that you had a smaller pool of protein to slow or delay disease progression?
Yes. Yeah, and so yeah, that's a lot to unpack. So so part of it is this RT quick that I mentioned. So 10 years ago, for example, like if you're on study section and you're reviewing grants, when there were prion disease grants trying to develop therapeutics, a lot of them would get shot down because, and the rationale, and I think maybe correctly, was we didn't have a means to diagnose prion diseases. So you have a disease that has 30 years of a subclinical phase and maybe a year of a clinical phase. And by the time a patient comes into the office with neurodegeneration, none of the drugs work at that point. Now with these ultra sensitive techniques is that there's work going on, right? And I think, like when I lecture to the medical students, I think the medical students of today, when they're practicing 5 to 10 years from now, there's going to be blood-based tests for these diseases that you can, pick somebody up 10, 20 years before onset. So now I think a lot of these therapeutics have a much better chance of working. And then to your question about, how do you direct the therapeutics? Yeah, I think that's the way to go, is you target the normal protein. So in PRP, so if you make a transgenic mouse or you knock out PRP, they're phenotypically normal, they're completely resistant to prion infection. And groups have made transgenic goats and transgenic sheep. And in Ames, I don't know if it's there anymore, there was some transgenic knockout cattle. It was funded by a pharmaceutical company. With the idea being is that if you were using fetal calf serum in making vaccines or something for human use, and how do you guarantee it's prion free? That's how you do it. So I think the difficult part, like when I have my hat on the ag and wildlife side, is that GMOs are socially just not, people aren't willing to accept that. So I don't see that being a viable mechanism of preventing like mad cow disease Or
Mhm,
with CWD, it's like you'd have to do a gene drive or something. Yeah,
mhm.
I just don't see that happening for politically. On
But
human.
I thought Huntington was, um, uh, you know, inherited, and there's a test for the mutation, and onset is like the penetrance of the disease is really high, and there's a population that they're going to die at a young age from this guaranteed. So the risk benefit is that, I
Yeah,
mean, maybe I'm
yeah,
wrong.
yes, there's working on that now. And in humans, there's actually a fairly well-known couple in the prion field that I guess probably in their late 30s now. Anyway, she has the mutation for FFI. And so when she found that out, she was a graduate student, and basically when she found out she was a carrier for FFI, went and trained in the prion field, and now she's working on developing means to lower PRPC levels for exactly what you're talking about, is for these familial forms where, yeah, I know I have this mutation and I have X percentage of chance of getting this disease by this age. Those are the populations that I think that will be targeted first for these types of treatments to lower down the normal precursor.
How similar are the different normal prion proteins from,
So,
you know?
yeah, so they're widely divergent, and so what I didn't have time to mention today, there's also another field they're called yeast prions. And so they're prions in the sense is, so yeast biologists think about them as an epigenetic mode of inheritance. So it's that conformational phenotype switch is a useful function for yeast. And so when the yeast geneticists get a hold of yeast prions, what they found is that it wasn't necessarily like a sequence that predicted kind of the ability to form a prion, but it was the amino acid kind of composition. So if you kind of look at tau and A beta and synuclein and PRP, they're wildly divergent, but all of them can have this kind of prion-like phenomenon. And I'm not enough of a biochemist to understand that at a really high level.
Really interesting.
Can I have another question?
Absolutely.
If no one else has one. Yeah, just about the correct, you mentioned that the public just doesn't accept GMOs. And this is all about science communication and elevating the public's knowledge about these things like GMOs and food irradiation, things that could save so many lives, but nobody goes down that road because they don't think anyone will buy it. So I just think maybe if we could think about ways to communicate your work in the prion disease research area, what types of things could we do if we didn't have to worry about that? And maybe slowly the public will change their mind about that if more people understood the potential for some of these things.
Yeah, and so there's a lot of work in this area with chronic wasting disease. So working with hunters, for example, and so I think that actually the Canadians do a much better job than we do with this. And the hard part is there's a lot of data suggesting when CW is first identified in an area, people are very concerned and want to have their deer tested and do X, Y, and Z. But then after 3 or 4 years, they just don't care anymore. So it's just it's hard to keep people's attention, and this thing with the gene drive, this was something that was proposed in Canada to try, and it's like, no way, you're not gonna, even though the technology exists, this isn't something that we want to do in a wild population.
Yeah, we're losing the battle of science communication because the anti-science communicators are more successful.
I mean, I think if there were, you know, some kind of genetic therapy for a disease with a really dramatic success, that might change things, but that's
Yeah.
tough.
Yeah, and I think this will happen with things like Parkinson's and you know, some of the human forms like this. And I think it's interesting going again to meetings in the different kind of worlds, is that the advocacy and the communication in like for Parkinson's is very different compared to PRP. So I do think it, you know, for some of the human diseases there is, you know, I think there's reasons to be hopeful scientifically and also socially.
I have a bit different question. So then it's well established now, yes, that the prion is a transmitting agent. And then Prusiner came up with this hypothesis that all these conformational change related diseases are related to prions, or the A beta and Huntington and so on are, or synuclein are prion proteins. But then are there any cases in where it's been shown that the Alzheimer's disease was obtained by transmitting of A beta protein? I mean the disease form or the other.
Yeah, there's a few papers, but you know, if it does happen, it's got to be super rare. And you know, so kind of my argument with this is that if you look at the classical prion diseases, both in humans and in animals, most of them are not contagious. So I don't use transmissibility as a definition for a prion. I think about it more as the yeast prion people do is this you know, epigenetic mode of inheritance. So I think in that vein, you know, a lot of these diseases kind of fit that model. And in some of the discussions that I've had with like the Alzheimer's field, I think the best argument for calling them like prion-like diseases and not prion disease is that because, as you point out, most people think that prions are transmissible and they're inevitably fatal. Is that going to affect how they get care? Are caregivers or clinicians going to not want to, you know, deal with patients because they think they might you know, get the disease? So again, that goes back to Dr. Keefe's question or comment about science communication. It's just we need to do a better job with that.
Yeah, but that goes back to the surgical equipment that if a surgery is being done on a Alzheimer's disease patient, then A beta got onto a surgical equipment. Can it be transmitted to other non?
Yeah, I'm not aware of any sort of surgical. The one that I'm most familiar with is with human growth hormone.
Yeah.
Is that there's some lots of human growth hormone that are contaminated with A beta, and some of the people that received that, you know, developed, you know, Alzheimer's disease.
Yeah, I remember that. Yes.
Yeah. And what's difficult about this, again, because these are prions and they don't have a nucleic acid genome, it's very correlative. It's very difficult to really, you know, positively link that source with the disease. You know, it's different than if I get the flu and I've been around a bunch of ducks. Well, you can sequence the flu that's swimming around in me and, you know, figure out exactly where it came from. But with prions, you don't have that. And that's one of the kind of scientific difficulties. When mad cow disease first came out in the human forms, I mean, it took about 3 years to do enough experiments to start to convince people that there was a link between the two. We just don't have very good tools for that.
Yeah, and then so the other thing is this, that we still don't know really what causes that the PRP-C changes its conformation going from C to SC.
Exactly. No idea.
And then the question I have, or suggestion or hypothesis or whatever, that so your different strengths may be related to different environmental factors. But what I mean environmental factor is not just outside, but within the body, the surroundings.
Yeah, and there's evidence for this in vitro systems where you can have prion formation occurring. If you put different cellular cofactors, you can start to change the strain properties.
Mm.
So I do think that the cellular environment, that there are cofactors that can influence which strain it is.
Yeah. Thank you.
Anyone else? Any more, Anne?
So when in these inherited disorders, yeah, sorry, you asked.
No, go for it. If you have time.
Or so just for, and this might be obvious and I just don't know, but are the inherited ones, do you think there's some kind of host susceptibility that's inherited that makes certain family members more susceptible to some common exposure? Maybe.
Yeah, I mean, the evidence right now suggests that. So again, you can do this in vitro systems where if you mutate the normal form and put in the familial mutations, is that it destabilizes the conformation of PRPC, and it just makes it more likely to want to misfold into that SC conformation. Now, whether or not there are kind of external events that might facilitate that, it's kind of like, with Alzheimer's disease for a long time, the number one risk factor was age. So with these familial forms, they tend to have a window of when the onset of disease occurs. And that seems to be very predictable, but whether or not it's due to any sort of, like in Parkinson's disease, I think there's a lot of evidence suggesting environmental factors may play a role. I'm not aware of anything. Having said that, I mean, again, there's so few familial cases of prion disease. I'm not sure anyone's done the proper studies.
I have one last thing. Given the nature of this conversation, interdisciplinary, and the size of our community within the Creighton School of Medicine and the university with all the, other health sciences, etc. Are there areas where you'd be interested in developing a collaboration with clinicians or, with people with whatever expertise?
Yeah, I think I'd be very interested in chatting with, the surgery department, and just again seeing what do they use for decontamination procedures. I think we could plug that into our systems to validate, what they're doing actually does what they think it's doing. I'm very interested in talking with neurologists again about these different protein misfolding diseases. Yeah, I'm collaborating with engineers and wildlife biologists already. So I think moving into kind of the more medical area makes a lot of sense.
Right, great. Well, you have a number of research leaders on this meeting, so we'll work on seeing what we can set up in terms of conversations.
I have a question.
Go ahead.
Yeah, it's Gopal from School of Pharmacology and Neuroscience. So does any host interacting partners are known for these prion peptides?
No. I mean, there was some studies about 15 years ago suggesting that PRPSC interacts with the laminin receptor. I don't think those have been validated, but no, there doesn't seem to be anything obvious. one kind of weird thing is that there's some evidence suggesting that PRPC might act as a receptor for tau and tau aggregates. And that PRPC seems to be important in kind of the neurodegenerative cascade in other protein misfolding diseases. So there's evidence that PRPC plays a role in neurodegeneration in Alzheimer's disease. But it's unclear exactly what that is. I mean, controversial.
Okay, thanks.
Hey, Jason, I got great talk. Got kind of a probably naive question. So with your now work in Parkinson's, and considering your past work looking at prions and their stability and how they react to different soils and everything. Same way, are you thinking at all like with alpha-synuclein, whether it can be, stable in the soil?
Yeah, I don't think so, in the sense of there's no evidence that synuclein is shed from people.
Okay.
So if you look at the classical prion diseases, one of the really weird things about chronic wasting disease is that deer, and for reasons we have no idea, deer shed a lot of prions in saliva and in urine. But like people, when people have CJD, they don't. So when I'm thinking about Parkinson's disease, I'm not really thinking about it as a transmissible disease. I'm thinking about it, okay, sometime in the brain, you get that self-templating form of the protein folding spontaneously. And then from there, it behaves like an infectious agent. But between people or interacting with the environment, yeah, I just don't think that's, I don't think that's, if it is a possibility, it's going to be so rare that to be non-existent. Otherwise, people like Dr. O'Keefe would be finding evidence for this.
Yeah. Okay, great. I'm glad I preempted that with a naive.
Well, I'm not sure we would find it because we wouldn't know to ask certain questions.
Yeah. Well, I mean, I was just thinking about it because, Nebraska, has a pretty high Parkinson's rate. And, you were talking before about prions kind of sticking to clay more so, and whether or not that could play a role at all.
Yeah, I mean, I think there's much stronger evidence for various pesticides and other things.
Yeah.
So if you think about what can make that normal form initially form into the self-templated conformation, it doesn't have to be protein, it can be something else. And I think that's where the Parkinson's field is asking, are there environmental things? Are there, something in the diet that could cause that initial seeding event? And then once that seed is formed, well, then it can self-template like a prion. But I think that initial formation could be at lots of things.
Yeah, in the Alzheimer's field, it was a hypothesis, and I think it still exists, the oxidative stress, which triggers the A-beta conformational change and then aggregation. So most likely for synuclein, this is the same case, or one of the cause, I think.
Agreed.
One thing that might shed a little light is if there were any occupational studies, like on maybe on Parkinson's, are there occupations that are higher risk or industries that are higher risk? Is it farmers who inhale a lot of dust, or people in certain occupations that might be at higher risk? I don't know if those studies have been done.
Yeah, the only ones I've kind of read myself, again, thinking about from like an infectious etiology, is family members. And there doesn't seem to be kind of an increased risk of family members. So you'd expect for the living in the same household for a long period of time, maybe that would, if there was some sort of transmissible, but that doesn't seem to be the case. So. which is,
All
consistent with- oh, sorry.
Go ahead,
Sorry,
Jason.
it's consistent with CJD because it's the same thing with the human prion CJD. Spouses and that don't get it as well, and it's because it's not shed. So
Oh, nice.
All right, well, it's a good thing we have one presentation today because we had lots and lots of questions and discussion. So I think we're out of time. Thank you again, Dr. Bartz, for presenting, and the rest of you for coming and the stimulating conversation. And I will see you next month,
thank
Or
you.
the month after.
Stay cool.